for your upcoming lecture exam, you should be able to draw pathways that would be involved in responding to a dropping blood pressure, responding to a rising blood pressure, and what the heart would be commanded to do as a result of that. I will also be drawing for you today the Withdrawal Reflex Arc and the Knee-Jerk Reflex Arc. We will also be drawing a reflex arc, or basically a pathway, I should say, for increasing gut motility prior to the left colic flexure, increasing gut motility after the left colic flexure, and how to draw a pathway to decrease gut motility. So there is a lot to draw today. Okay, well, the first picture I'm going to draw for us is going to be how we decrease gut motility. Now, which division is in control to decrease gut motility? Any ideas?
Sympathetic.
It is sympathetic, which is why I have a picture of the spinal cord over here on the left and the sympathetic trunk. But I also have a picture of a brain. And the idea is some students would have said it's parasympathetic, and they would have started with the brain, and they would have started drawing cranial nerve number 10, which innervates down into the gut. I'm going to actually copy this slide. That's not what I wanted to do. Copy. Paste. There we go. That's what I wanted. Okay, so as I said, let's start with decreasing gut motility. So if we want to decrease gut motility, this is going to be a sympathetic pathway. I have more than one recording activity happening right now, so my computer is running a lot of programs right now. Just going to close some extra ones. There we go, that should help. Okay, that should help. Okay, so we need to draw a 2-neuron chain to decrease gut motility. The sympathetic division is thoracolumbar, meaning that's where we find the preganglionic sympathetic cell body. It's found in the IMLCC at levels T1 through L2. Now, our gut, all parts of our gut, happens to be below the diaphragm. And last week, we talked about options that the preganglionic and postganglionic fiber or axon had as part of these pathways that I said we would draw again today. So the preganglionic cell body is found in the IMLCC at levels T1 through L2. That first axon would exit through the ventral rootlet, enter a ventral root, enter an anterior primary ramus, and then it would enter a gray ramus,
Is that a gray? The one on the left towards the top, that's a white ramus or a gray ramus? I thought the ones on the outside were white.
Sorry, white, yes, white.
Okay.
The postganglionic
Thank you.
fiber is gray. Yes, sorry.
Got it. Okay, that's okay. Thank you.
Thank you. So because we are going to structures below the diaphragm, we are going to use a collateral ganglion, also known as a prevertebral ganglion. And if we want to be more specific, it would be the celiac ganglion, superior mesenteric ganglion, or inferior mesenteric ganglion. The postganglionic cell body is found in that collateral ganglion. The fiber would then terminate. I'm just going to draw that it's terminating on the stomach. I could have drawn it to any part of the gut. So I'm just drawing it to the stomach. And on your exam, I will also ask you to label these structures, meaning where is the preganglionic fiber, where is the postganglionic fiber, because students draw these things and then they don't label them correctly, and then that they lose points for that. So in green, I have the preganglionic fiber. The cell body, like I said, is in the IMLCC. In blue, I have the postganglionic fiber with the cell body in a collateral ganglion. Now, also on your exam, you should be ready to identify what neurotransmitters are released from the preganglionic fiber and the postganglionic fiber. So the preganglionic fiber would release acetylcholine. I'm going to write that in green. And the postganglionic fiber would release epinephrine. And I'm writing that in blue, just so you know which neurotransmitter goes to which fiber. I'm going to keep labeling some other structures that are shown on this picture just as review. Up here is the dorsal gray horn or column. Down here would be the ventral gray horn or column. This structure that I'm highlighting in blue is the sympathetic trunk. And the structure that the green preganglionic fiber is coming through is a white communicating ramus. The other one, which would be an exit potentially, depending on what kind of reflex arc we were drawing or pathway, is a gray communicating ramus. So the white communicating ramus is the entrance into the sympathetic trunk, and the gray communicating ramus is the exit out of the sympathetic trunk. All right, and then these structures here that I'm highlighting in orange along the sympathetic trunk, those are sympathetic ganglia, also known as paravertebral. Para means next to. Are there any questions about that? Okay. Same picture. Now I am going to show you increased gut motility, which would be parasympathetic. And parasympathetic division is also known as the craniosacral division, referring to the two locations where you would find preganglionic cell bodies. You would find them either in the brain for cranial nerves 3, 7, 9, or 10, or you would find them in the sacral spinal cord at levels S2, S3, S4. Let's do increased gut motility prior to the left colic flexure. Where is the left colic flexure? The left colic flexure is referring to the large intestine, which I'm coloring here in blue, and the left colic flexure, flexure means bend, is over here where we see this turn of the large intestine turning close to the stomach. So I'm going to mark here. This part is called the ascending colon because it's going upward towards the head. This part is called the transverse because it's going horizontally. And then this part down here is the descending colon. So that's the large intestine. This structure over here, just give me a moment, that I'm drawing in yellow, that's the right colic flexure. So right colic flexure and left colic flexure. Now, why is the left colic flexure an important landmark? Why am I bringing this up? If you were to eat food, if you're looking at this picture, when you swallow, that food would enter your esophagus, then it would enter your stomach, then it would enter your small intestine. There are three parts to your small intestine: the duodenum, the jejunum, and the ilium. After the ilium, and you can see here the ilium, I wrote over it, so let me erase that. The ilium is this last part The ilium is this last part of the small intestine. And from the small intestine, your food would enter your large intestine that is highlighted here in blue. The food would enter the small intestine to the large intestine, and after it passed into the large intestine, we no longer call it food. We would call it poo. We would call it fecal matter. Your fecal matter would go upward, up through the ascending colon, then go from right to left through the transverse colon, and then it would go into the descending colon, and then it would enter the sigmoid colon, and then out your anus. Cranial nerve number 10, the vagus nerve, is part of the parasympathetic division. It is the only cranial nerve that descends not only into the thoracic cavity, but also into the abdominal cavity. It is a preganglionic parasympathetic fiber. It has postganglionic partners that would innervate the smooth muscle found in your gut. That's what we're talking about right now. And if the parasympathetic division has more control, you would have more gut motility in these structures that you're seeing in the picture on the right-hand side. But the cranial nerve number 10 does not reach all the way down past the left colic flexure. It would not tag team with postganglionic fibers past the left colic flexure. We would need the sacral spinal cord to be involved if we wanted to target increased gut motility past distal to the left colic flexure. And I will go over that in just a moment. So let's draw our preganglionic fiber now. I'm going to use a different color. I'm going to use purple for my preganglionic fiber. Cranial nerve 10 has nuclei in the brainstem, so I'm drawing that here in magenta color. Let's say we want to increase gut motility of the stomach. It, of course, would extend from the brain, down the neck, into the abdominal cavity. It would synapse on a postganglionic fiber very close to the target organ. Remember, for the parasympathetic division, there is a very long preganglionic fiber, very large in length, and a very short postganglionic fiber, because the postganglionic cell bodies are found in the wall of the involuntary effector. So I'm going to draw in orange the postganglionic cell body, and it terminates on the smooth muscle on the stomach. That's what I'm drawing here. You might be asked to tell me what neurotransmitters are released from the preganglionic fiber, which is drawn in magenta or purple. You would have acetylcholine being released. And from the postganglionic fiber, we would also have acetylcholine being released. Are there any questions about increasing gut motility prior to the left colic flexure? Okay, well, let's move on to increasing gut motility after the left colic flexure. So this is a picture in the PowerPoint. I just rearranged it to bring it more closer to the beginning of the PowerPoint, but it is in the PowerPoint if you've already printed it. I think it was slide 7 at one point. I just moved it up. Now we want to increase gut motility after the left colic flexure. Distal to the left colic flexure. Now we would need our spinal cord picture. We would not be using a cranial nerve from the head. We would not be using cranial nerve number 10. Let's say we're trying to increase gut motility of the descending colon. And I'll remind you, right here is the left colic flexure. So if I want to increase gut motility for the descending colon, I need to use my spinal cord picture. This means I'm using a spinal cord picture that would be at levels S2 through S4. Structures that are shown here in this picture would not be present at this level. So I'm going to cross them out because they would not be there. For example, the white communicating ramus would not be there. White communicating rami are only found at levels T1 through L2. So that structure that I put a purple X through on both sides would not be present. Just to orient you, this structure here is the dorsal root ganglion. There's one on this side as well. This would be a posterior primary rami. And this is an anterior primary ramus. Of course, there is one on the left and right. This structure here that is found on both sides is called a pelvic splanchnic nerve. I'll remind you from last week that splanchnic nerves have preganglionic fibers in them. So our two-neuron chain would begin with the preganglionic cell body found in a lateral gray horn. At this level, the lateral gray horn is called the sacral lateral gray column, SLGC. It would exit through a ventral rootlet into a ventral root. It would enter an anterior primary ramus for a short time. It would exit through a pelvic splanchnic nerve, and it would get very close to its target organ before it synapses on the postganglionic cell body, which would be found in the wall of the involuntary effector. What neurotransmitters would be released from each neuron, or at the terminal bouton for each neuron that I've drawn? Again, acetylcholine for the preganglionic fiber and acetylcholine for the postganglionic fiber. Are there any questions? Okay. Well, now let's talk about how we would increase or decrease heart rate. Which division would increase heart rate?
Sympathetic.
It would be sympathetic, correct. All right, so just to pull from our lecture unit 2 material when we learned about baroreceptors, I'll remind you that baroreceptors are found in the aortic arch and in the carotid sinus. They are modified nerve endings. They send sensory afferent signals up to the brain. So let's say the person has a declining blood pressure. So I'm going to draw a reflex arc for declining blood pressure. In my picture, I'm going to put this squiggle line here. I'll just make a big dot. How about that? So here's my legend. The baroreceptor that I'm drawing is in the aortic arch. If blood pressure is dropping, the the afferent fiber would send information up to the medulla oblongata. Which nerve is actually sending this information up to the medulla oblongata? It's actually cranial nerve number 10, the vagus nerve, and I'll remind you that it has an afferent branch to it, a sensory branch. So cranial nerve 10 is sending up action potentials to the medulla oblongata. These action potentials would not be very frequent. There would be very few of them reaching the medulla oblongata. Fewer action potentials to the medulla oblongata tells the medulla oblongata that blood pressure is dropping. The medulla oblongata is the integration center. What appropriate output command should we send to the heart? And the answer is increase heart rate. This will help bring blood pressure back up to where it should be. Well, that is a sympathetic division now, a sympathetic division response. So how can we draw this? Well, of course, the medulla oblongata would send action potentials down through the spinal cord until it got to levels T1 through L2. Obviously, that's where the sympathetic division is found. So the preganglionic fiber would exit through a ventral rootlet, enter an anterior primary ramus for a short time, enter a white communicating ramus, and it would synapse in the sympathetic trunk on a postganglionic cell body. Now, what would the postganglionic fiber do? It is not going to go into a collateral ganglion. Those only exist below the diaphragm. The heart is obviously above the diaphragm. So a collateral ganglion and a splanchnic nerve would not even be at this level that we're entertaining at this point. So those structures would not even be available to you. In your homework packet, you were asked to draw an X through some structures that would not be applicable, that were drawn on the picture, but you were supposed to draw an X through them to show that they weren't applicable to the task you were being asked to do. That's why I'm crossing out the collateral ganglion. That is only found below the diaphragm, and we are stimulating an organ that is above the diaphragm. The postganglionic fiber actually exits the sympathetic trunk. It does not go through a gray communicating ramus. And it would synapse on the sinoatrial node of the heart. And it would increase the heart rate. What neurotransmitters are released from the pre and postganglionic neuron? From the pre, it's acetylcholine, and from the post, it would be epinephrine. Are there any questions? Okay, well now let's draw the reflex arc for increasing blood pressure. Again, here is my baroreceptor in the aortic arch. Cranial nerve 10 has afferent pathways, or I should say sends afferent signals up to the medulla oblongata. All of that is still the same. The number of action potentials would actually be increased. There would be an increased rate of firing. More action potentials arriving to the medulla oblongata tells the oblongata that blood pressure is too high. The medulla oblongata is the integration center. What outgoing command should it send? If we want to lower heart rate and lower blood pressure, this should be a parasympathetic division response. Cranial nerve 10 has preganglionic cell bodies in the medulla oblongata. It would come down the neck into the thoracic cavity and get very close to the wall of the heart. It would then synapse with its postganglionic partner within the wall between the epicardium and myocardium. And then the postganglionic neuron would innervate the sinoatrial node, and it would decrease heart rate. What neurotransmitters are released from each of these neurons? From the preganglionic fiber, it would release acetylcholine, and from the postganglionic fiber, it would also release acetylcholine. We would not draw anything in the sympathetic trunk because a rising blood pressure would lead to a decreased heart rate. Decreasing heart rate is not a function of the sympathetic division. Are there any questions? Okay. Well, then we already did increased gut motility below the left colic flexure. I just duplicated an extra slide. We also did increased gut motility prior to the left colic flexure. We also did decreased gut motility using the sympathetic trunk. This picture here came from your homework packet, and I included it here to remind me to tell you that in your homework packet, it asks you to identify the structure that the letter X is on, and yet the letter X in your homework packet has been moved. It doesn't land on anything in your homework packet. Perhaps you've seen that. Well, the letter X is supposed to be on this structure here, and that is the anterior primary ramus. Okay, now we are going to do the Patellar Reflex Arc. And in order to do this reflex arc, I am using this generic picture that is sadly drawn, a transverse section through your spinal cord. I'm going to draw a person's knee and foot. And their thigh. So here's their kneecap and a very sad, poorly drawn foot. I'll remind you that the muscle group on the anterior side would extend your lower leg, and those are your quadriceps muscle groups. On the posterior side, we have the hamstring muscle group, which would flex your lower leg. If we are doing a patellar or knee jerk response, we would want to stimulate the quadriceps but inhibit the hamstrings. What would this look like? First of all, it is not a two-neuron chain. We're not worried about a preganglionic and a postganglionic neuron. That only applies to the sympathetic and parasympathetic division. We're targeting skeletal muscle, which means we use a somatic motor neuron. Let's draw the neuron that would innervate the quadriceps muscle group. So we have the somatic motor neuron would be in the ventral gray horn. It would enter a ventral root. It would enter an anterior primary ramus. And this neuron would synapse on the quadriceps muscle group. What neurotransmitter is released? Acetylcholine. So I'm just going to write acetylcholine next to it. Now, what started this reflex arc? Well, when we're talking about the knee-jerk reflex arc or the patellar reflex arc, we think of our doctor with the little rubber hammer hitting our patellar ligament. When it hits our patellar ligament, it causes our quadriceps muscles to stretch. Momentarily and very quickly, our quadriceps muscle group is stretched. Muscles can rip off your bone if they are overly stretched. As a result, the reflex that muscles will use is to contract. Think about that. If a muscle is stretched too much, the appropriate action would be to contract, to shorten. That's an antagonistic reflex, right? If it's elongated and could rip off of the bone, then the correction is to shorten, to contract. And in this case, when the quadriceps contract, the lower leg is extended. So this afferent fiber is actually found in the quadriceps muscles. There are special receptors in all muscles. Any muscle that can be stretched has these special receptors. They're called spindle fibers, if you want to know what they are. So I'm going to draw a spindle fiber here in blue. And I always tell my physiology students, think S for spindle and S for stretch. So these receptors respond to too much stretch, and they send afferent action potentials up through an anterior primary ramus into a dorsal root. The cell body of this afferent fiber is found in the dorsal root ganglion. And in your textbook, this is called a monosynaptic reflex arc because this afferent fiber that I've drawn in blue will terminate on the somatic motor neuron that I've drawn here in purple. And it will stimulate that purple neuron, that somatic motor neuron, and it will tell the quadriceps muscle group to contract when it releases acetylcholine.
What's that called again?
Which one? What are you talking about?
The single single motor, that where this the spindle hooks back up with the original.
You mean this area right here?
Yeah.
Well, that's a synapse.
No, but you said it's called like a single or a mono or a
monosynaptic reflex arc.
That's what I was asking for. Thank you.
Yeah. But it's actually misleading, Jessica, because this afferent fiber, it's called monosynaptic because it only has one synapse between the afferent fiber and the somatic motor neuron that would activate a particular muscle group. In actuality, that afferent fiber would also synapse on a different somatic motor neuron. It would also synapse, I shouldn't say on a somatic motor neuron, it would also synapse on an interneuron. And that interneuron would in turn synapse on a somatic motor neuron that innervates the hamstring group. I'll remind you from lecture unit 1 that somatic motor neurons only release acetylcholine. And if they release acetylcholine, then the skeletal muscle they innervate will reach threshold, and that means generate an action potential, and then that muscle group will contract. When you're having a knee-jerk reaction, you want the quadriceps muscle group to contract, but you don't want the hamstring group to contract. In the lecture, you saw for this reflex arc that the afferent fiber, the spindle fiber, releases an excitatory neurotransmitter. And in the lecture, I had a plus sign to designate an excitatory neurotransmitter. Acetylcholine is considered to be excitatory. But it's not the only neurotransmitter that can be used for excitation. So I just put a plus sign to show that this spindle fiber would cause the afferent nerve to release excitatory neurotransmitter on these neurons that are shown in the spinal cord. That means in purple, the somatic motor neuron that innervates the quadriceps would reach threshold and would fire an action potential, releasing acetylcholine on the quadriceps. They would contract and you would get your knee-jerk reaction. But the interneuron that I've drawn here in orange would also reach threshold. It would fire an action potential. But what kind of neurotransmitter would this interneuron release onto the somatic motor neuron shown here in green that innervates the hamstrings? It would release an inhibitory neurotransmitter. So I'm showing this as a negative sign. Negative sign means inhibitory neurotransmitter released on the green somatic motor neuron. If the green somatic motor neuron receives inhibitory neurotransmitter, it would hyperpolarize, become more negative. It would not reach threshold. It would not fire an action potential. That means the hamstring muscle group would stay relaxed. We would want this to happen because the hamstring muscle group is an antagonistic muscle group to the quadriceps. If the quadriceps are contracting during this knee-jerk reaction, we would want the hamstring muscle group to be inhibited, to stay relaxed. Are there any questions?
Kara, for our test, if this is on this, are you going to want us to have like different colored pencils so that you can see it easier?
That is always helpful. However, you're going to be asked to label things. So not everyone has different colored pens or pencils. If you label your neurons, then I could figure out which one's your afferent because you're going to say afferent. And I could figure out which one is your somatic motor neuron that would be activated.
Okay.
So I've been giving this exam for many, many, many years. And trust me, I have ways to make sure, even if you only have a pencil, you can still draw this and I can still figure out what it is that you're identifying. So again, the green is showing you the somatic motor neuron to the hamstrings, which should stay relaxed. And again, in orange, I'm showing you the interneuron that would release inhibitory neurotransmitter on the somatic motor neuron that serves the hamstrings. That somatic motor neuron that serves the hamstrings would be hyperpolarized. It would not reach threshold. That means the hamstrings would not be striking an action potential and then contracting. So this is our knee jerk or patellar reflex arc. In your lecture, I do show you a nice picture of the knee-jerk reflex arc that has all of these neurons labeled. It also uses pluses and minuses to designate an excitatory neurotransmitter versus inhibitory. I think that was in the peripheral nervous system lecture. I'm just checking that real quickly. Yes, on slide 13 of your peripheral nervous system lecture shows you the knee-jerk reflex arc, all labeled, pretty little colors. On the left side, picture A of slide 13, it shows you the initial stimulus, the afferent signal. And then on the B side, on the right side of that slide, it shows you the response with the quadriceps muscle group contracting, and you see the person's lower leg extend. On slide 14, there is the withdrawal reflex arc. I'm just going to copy that slide real quickly and bring it into our pathways picture. So right now you all should be seeing the withdrawal reflex arc from the peripheral nervous system lecture. I could draw it here on this picture of the spinal cord, but it gets very messy. So instead, I think I'm going to talk through this picture that is, again, from slide 14 in your peripheral nervous system lecture. In blue, you're seeing the afferent fiber. So the withdrawal reflex arc is also called the crossed extensor. Please notice in this picture we have the right arm, and in this picture the right arm is the one that's being pulled. It's being hurt. Sometimes in class, instead of showing someone's arm being pulled, I demonstrate someone's right foot stepping on a piece of glass. Whichever limb is being pulled or hurt, that's going to be the limb that's going to pull away. If you're being hurt or cut, you withdraw your hurt limb. You bring it in close to you. You want to protect it. That's why this is called the withdrawal reflex arc. But the other side of your body extends. So if someone actually comes up and tries to pull you, let's say they're trying to kidnap you or abduct you, and they pull you by your right arm, trying to pull you into a windowless white van, you're going to instinctively pull your right arm back to you, and with your left arm, you're going to push against your assailant. I once took a self-defense class, and our instructor said that when someone grabs you like this, here's your reflex. You instinctively want to pull away with the arm that the assailant is grabbing, and the opposite arm pushes away. A lot of assailants anticipate this, and they brace themselves for this reaction. My self-defense instructor had to teach us, we had to retrain our brain to override this actual reflex arc to catch the assailant off guard. I have to tell you, you can't practice this enough. Your instinct, your reflex is very hard to override. It's very difficult. So again, whatever limb is being hurt or pulled or threatened in any way, that's what you instinctively bring back closer to your body to protect. And the opposite arm or opposite leg, the opposite limb, pushes away. That's why this is called the withdrawal reflex arc or the crossed extensor reflex arc. Hurt limb close, non-hurt limb extends, pushes away. You want to remove yourself, push away from the hurtful stimulus. So again, in blue, we see the afferent fiber. It travels through a dorsal root. We can see the cell body in the dorsal root ganglion. And in green, we're seeing interneurons. And we're seeing a lot of interneurons here. We see this first interneuron. I'm going to label it as number 1. We see this first interneuron making synapses on other green interneurons. These other green interneurons are both across the spinal cord on the other side. That's called contralateral. And there are also green interneurons that are on the same side. That's called ipsilateral. So here are 2 interneurons that are ipsilateral. I'm just going to put ipsi. And here are some interneurons that are contralateral. They're on the opposite side. In purple, you're seeing an efferent somatic motor neuron that innervates the triceps. So I'm going to write that. There is a somatic motor neuron innervating the triceps on the right arm and on the left arm. In red, we're seeing the somatic motor neuron that innervates the biceps. Somatic motor, I'm just going to put SMN for somatic motor neuron, innervating the biceps. Notice for the right arm, which is being hurt, which is being grabbed, we want the interneuron to release an excitatory neurotransmitter. Look up here. The somatic motor neuron innervating the biceps on the right side is being stimulated, reaching threshold. It would fire an action potential and stimulate the biceps muscle group. This would cause flexion of your forearm. You would withdraw from the hurtful stimulus. But notice that the somatic motor neuron that innervates the triceps on the right side is getting an inhibitory neurotransmitter from the interneuron, shown up here with the negative sign. That means that somatic motor neuron innervating the triceps would be hyperpolarized, not reach threshold, would not fire an action potential, and the triceps would stay relaxed. The triceps are an antagonistic muscle group to the biceps. If the biceps are contracting, the triceps should stay relaxed. But what is happening on the left side? Again, this reflex arc is also called the crossed extensor. So on the left side, the person wants to push away from their assailant. Notice that the somatic motor neuron on the left side that innervates the triceps, it is receiving an excitatory neurotransmitter. It would reach threshold, it would spike an action potential, and it would stimulate the triceps muscle group. They would contract. If the triceps contract, you extend your forearm. If the triceps are contracting, then the biceps should stay relaxed. Notice that the interneuron on the left side is releasing an inhibitory neurotransmitter on the somatic motor neuron that innervates the biceps. Again, the biceps should stay relaxed if the triceps are contracting during this extension action. Are there any questions about this? All right, just so that it's a nice picture. I'm going to copy and paste the knee-jerk reaction picture into this PowerPoint, and I will re-upload this Pathways PowerPoint with my notes included onto our Canvas shell under Lecture Unit 3. So you can have the blank template to practice with, and you can also see my notes to also refer to. Are there any questions? I'm saving this before I forget.